as I mentioned, when you were listening to that, watching the raw video, and I was pointing out, they're really like these two modes. There's the rhythmic mode, where you hear this this theta oscillation, and then there's this bursting mode. And those two modes, the rhythmic and the bursting, which is illustrated here, this is like local field potential, kind of macroscopic electrical activity. So we're not looking at single cells, we're looking at the relative modulation of large populations. This is sometimes you might think of it as like the EEG, which would be like a surface version of this. But what you see here is that you get this population modulation. This is the rhythmic modulation known as the theta rhythm, about a 10 hertz oscillation. When the animal stops within about a fraction of a second, you get this transition between this aperiodic bursting, these large sharp deflections referred to as sharp waves in the hippocampus because of their the morphology here. And then if you looked at it in more detail, you would see that on top of these sharp waves, there's a high frequency oscillation referred to as a ripple. So these are referred to as sharp wave ripple events. You can't see the ripples here just because it's old and the filtering kind of took that out. But you have these two modes. And what's interesting is that the theta rhythmic mode is expressed when the animal's moving, that's sufficient to drive it, but can also be expressed when the animal's stationary but attentive. And there's some interesting differences. Classically, they were referred to as two different types of theta, type 1 and type 2. Type 1 and type 2, they had different dependencies, sensitivities to acetylcholine, cholinergic modulation. They were slightly different frequencies. They were driven by different input systems. But the idea was, when you're taking in information from the world, either by moving or attending, you turn on this rhythm. There might be slight dependencies on the frequency and other thing, but this is the online mode. This is when you're taking information in. When you're inattentive, and that is either when you're asleep, you see this when you're asleep, or if I'm active, I'm moving, but inattentive. Things like feeding, grooming, copulation, anything where I'm taking actions. It's not like the amount of, you can be very active. It's just that I don't have to pay attention to the outside world, right? Think of these as like reflexive responses. I don't have to be thinking about what I'm actually doing. I can just be responding. In that case, the hippocampus can switch off into this offline mode, but it's not off in the sense that it's not active. It's just active in this different way. So this is taking information from the outside world. This is now in this quiet, wakeful, or offline state. That's the way I refer to it. So focus on both of these. The online state, this is the theta rhythmic mode. When you look in detail at activity with respect to this rhythm, the theta rhythm, you see something very interesting. And this is something that John O'Keefe discovered. He discovered place cells in the 1970s. He discovered this property known as phase precession in 1990, 91. And what John noted was that when an animal is moving through a place field, not only does the number of spikes, the firing rate increase, but the precise timing of individual spikes with respect to this underlying theta rhythm, it changes. And so here, this is in time, but this is an animal that's running through its place field. As it's running through its place field, the number of spikes is increasing, the firing rate is going up. But if you look at the timing, the first spike occurs here at about the peak, and then as more and more spikes occur, the first spike is earlier and earlier, and that is that the phase advances as the animal moves through the place field. Now, there are two things that are actually illustrated here. In red, this is spiking activity in the hippocampus. This is the theta oscillation measured in the hippocampus. And up here, this is actually activity measured in the prefrontal cortex. Completely different structure. It is connected, the hippocampus, in particular the ventral hippocampus, talks to the prefrontal cortex. But looking at the local field potential, I don't see the theta oscillation. Again, the prefrontal cortex, it's not necessarily coding place, but if you look at the timing of spikes in the prefrontal cortex, they're phase locked to the hippocampal theta oscillation. So the other principle that you can see illustrated here is the idea that the theta rhythm can serve not only as a mechanism, because you have a regular oscillation, you can look at the timing of spikes with respect to that oscillation, or it gives you another dimension, phase, to do coding, but the fact that you have this oscillation means that you can actually use this to couple or to coordinate the communication through the coordination of rhythms across distance. You can think of as using a coherence signal. So you can use two properties, phase and coherence, to both code and communicate. So now this rhythm, so you can say, okay, so I got this rhythm. When I'm running, I can use like coding, whatever this coding is, and then I can also use this to like communicate that code to the prefrontal cortex. Now the interesting thing about the coding or the coherence and communication is that you can do this dynamically. In other words, hippocampus can talk to the prefrontal cortex. If I just have a synapse, a wire, that wire is always present. Maybe the prefrontal cortex doesn't want to listen to hippocampus. Well, what does it do? It can't like cut the wire. And so what it can do though is if I break the coherence, then I can ignore the hippocampus. And so I can dynamically or selectively engage communication with the hippocampus by selectively establishing coherence with this rhythm. Now we can talk about how does the hippocampal theta rhythm come to influence activity in the prefrontal cortex. And so there's been a lot of work on that. I can say, it can either be through the direct connection, the excitatory connections from the hippocampus driving prefrontal cells, or inhibitory connections. That is, inhibitory cells in the hippocampus modulating excitability in the prefrontal cortex. And the inhibitory modulation is the sort of the preferred model, either directly or indirectly, excitatory cells driving inhibition. So inhibition controlling the dynamic modulation of excitability of the rhythm, and then the excitatory connections communicating the information. So those two ideas, coding, communication, or coding, coherence, or like phase coding, coherence and communication, and the idea of excitatory and inhibitory communication and regulation of dynamics is really expressed here in this sort of it's a cartoon that illustrates the property of what I referred to, what I showed in the last slide, of phase precession. And that's the relationship between position and phase of theta oscillation. Now this is like real data. So this is, in an animal, we've taken one place cell, and then with repeated passes through there, mapped out the relative phase in individual points or individual action potentials. So when does that spike fire with respect to the phase of that theta oscillation? And what you see here is that as the animal enters the field, spikes start off late in phase, and then as the animal moves through the field, it advances and gets earlier and earlier. And so when John first published this, he referred to this phase precession. And so this is what I was talking to you earlier about when he noted this. So if you just look at like firing rate, like the cell was firing, you can tell, the animal was like in this location, right? You get about, okay, so it's maybe like, one place cell, 40 centimeters. But if I use phase, if I look additionally at the phase, I can tell, if he's in the early part of the field or the late part of the field, I get very different phase profiles. And so the idea was this phase can be used to give me not only where he is, but the relative position within the field. This was the place code. And then the firing rate, he argued, was the velocity. But there are a couple of other interesting things to note when you look at this. And that is when, this is sort of first published, and there was the argument that phase coding represents a 360-degree shift, right? You move from 0 to 360. This is the phase precession curve. But if you actually look at it, many of the cells show this. It's not strictly linear. It shows this kind of banana shape, right? Where if you look here in the early part of the field, if you kind of ignore this, the later half, first part of the field, you get pretty good. There's pretty good linearity. I mean, the correlation between position and phase is pretty good. If you just ignore, if I ignore this, you look at the latter half of the field, it's pretty lousy, actually. There's, and that's because there's a lot of spikes, many spikes here, high firing rate, poor phase coding, low firing rate, good phase coding. So you can think of there being like early part of the field, late part of the field, two different kind of coding properties. But you can also do that if you separate here in phase. Late phase, this part, good linearity. If I look at the early phase, crappy phase coding, right? This is poor. So if I look at the early phase, If I look at early phase of firing, this place, it basically doesn't even fire over here. If it fires, what I can say is with good confidence, yeah, the animal's probably over here, right? Based on the number of spikes. So I can't, I can use the rate code. So the rate code is effective in giving me a static estimate of likelihood that the animal is in this place. Whereas the phase code, there are fewer spikes, the firing rate is lower, but I can use the phase of that to give a much higher resolution estimate of the position within the field. So there are these, like, think of there like these sort of the 4 quadrants. There's the early field, early phase, late phase, different coding properties. And on the left is just like a simple model. This was when Mayank Mehta came up with this model for how could you get this phase precession using a very simple biophysical model. And in the simple biophysical model, what you have is excitation in blue and inhibition in red. And so excitation, you think of it, the excitatory input as an animal moves through the field, the excitatory input changes in a graded fashion. Now that graded input change could come from, sort of many modalities that are modulated by let's say a distance metric. As I get closer to the podium, the thing gets larger. So you can think of there's lots of gradients of things in the world. Sounds get louder as I get closer, all of these things. And so the idea that the world, the input space, has a natural gradient structure. Things are going to change as a function of position, because that's the way they are in the real world. The real world actually does have inputs that change in a smooth fashion as I move through space. So you think of gradient organization of the inputs, and then in red, you have the ability to dynamically modulate activity arbitrarily, and you can do that through inhibition. So, and one thing about inhibitory cells relative to excitatory cells is that they show this very strong theta rhythmic modulation. They fire at very high rate. So they're really good, lots of spikes, firing in this rhythmic fashion. It would be great for actually imposing this background variation in excitability. So if you think about the simple model, inhibition is varying in time, excitation is varying in space, and it's graded, and now you combine the two and you introduce this very simple biophysical premise, which is that a spike will be produced when excitation exceeds inhibition. That's all. So whenever the blue line is above the red, you're going to get a spike. And so what you see is that when excitation is low, you have to wait until inhibition gets all the way down here to produce a spike. As excitation increases, you can fire earlier and earlier. But in addition to firing earlier and earlier, the amount of time the blue line spends over the red line increases. So this might be the timing of the first spike, but you can get spikes in here as well. And so you're going to get more spikes late in the field, more spikes with more variable phase. They can fire anywhere in this region. And so it captures both this phase advance, also the phase variability that you see in real phase, in phase precession curves. And so the idea is that this biophysical property could give you this both the kind of the temporal and the rate coding properties you see in natural cells. Now, okay, so but there are lots of ways you could get that. But so what? Phase coding, phase precession rate. How does this relate to any things that we were talking about before? In particular, this idea of episodic or sequential like event coding. And that comes up when you now introduce a second cell. So now we have 2 place cells, 2 place fields. Place cell 1 has a place field that's to the left of place cell 2. Both have this kind of graded, the inputs are graded. But now what you see is that when I introduce this, so place cell 1 as this, spikes fire late, earlier and earlier, place cell 2, same thing. Place cell 1 is always going to fire before place cell 2 because the input to 1 is always going to be stronger than 2. And as if I have gradient input structure, I'm going to get this kind of phase coding that will give me a natural sequence, 1, 2, 1, 2, 1, 2. So every theta cycle, I will express the distance between these 2 place fields as a phase shift here in the theta cycle. And that is, I transform this, what I refer to as behavioral time scale. Why do I refer to it as the behavioral time scale? Because this is separation in distance. How long does it take the animal to go from 1 to 0 to 20 centimeters? I have no idea. Depends on how fast it's running. If it's running fast, it could be, could be a second. If it's slow, it could be 5 seconds. And so the idea is that its behavioral timescale is long and variable. Biophysical timescale, on the other hand, I can actually control by controlling the frequency of this oscillation, and it's fixed. It's short and it's fixed, and it's under my control. And so what this would really be doing is it would be coding behavioral sequences that are dependent upon locations changing over time, but eliminating mapping variable behavioral time, long variable behavioral time scale into fixed biophysical time scale. Now, why would fixed biophysical time scale that captured order be relevant? Well, this is now, we go back maybe even longer, almost 40 years, into these models of synaptic plasticity and learning. There was the so-called Hebbian notion, which is classically conceived of as like it's the sort of the fire together, wire together. The idea that inputs that are concurrently active should be strengthened. The actual model was one in which inputs or outputs that are predicted by an input the input should be strengthened. And so the idea was that there should be a causal ordering to that, inputs that predict output. And this was demonstrated explicitly by looking at what's so-called spike time-dependent plasticity. And that is if you activate the output of a cell, the inputs that are active before that output are strengthened. The inputs that are active after that output are weakened. This was something that had been demonstrated by Mu-Ming Pu and Henry Markram kind of independently in different preparations. But this idea that the timing actually matters. And so that what you really want, you don't want to know whether or not place cell 1 and place cell 2 were firing together. What you really want to know is did place cell 1 fire before place cell 2, because that gives you like the behavioral trajectory. It says, oh, you're going in this direction, which means by having that causal ordering, it allows you to estimate, well, where are you going to end up next? If I have this ordering, 1, 2, I can say where I'm going to end up next is I'm going to end up over here. And so I can use this, the causal estimator, to do predictive estimation. So you say, I use the past, where did I come from, where am I now, to predict where I'm going to be in the future. And so if we now take this kind of simple idea, I can use the ordering, and now I go back to the real data. I use this Bayesian decoding that I showed you before, where I got the little triangles showing the position, but now I'm going to do it at higher resolution. So that, that movie was done at like 200 millisecond resolution. I was doing it was like 5 times a second, I'm estimating position. Well, if I take that down now to 20 millisecond resolution, which means I can now estimate position at a sub-theta cycle time scale. So instead of 200 milliseconds, 20 milliseconds, and so I can now decode position within a theta cycle. And when I decode that position within a theta cycle, so it's now time, I'm doing decoding, the gray boxes are like little triangles. Now I see, and this is the animal's actual trajectory, now I see, it's not that the gray estimate just like, kind of covers the animal's location. What you can see is that it sweeps from just behind the animal to just in front of the animal each and every theta cycle. So the real hippocampal code is not just a positional code, where am I now? It's like, where was I and where am I going? Past to future, 10 times a second. This is what I'm getting. And so now if I take all of these, I lock them to the peak theta oscillation, I average them. Now I say, okay, what's the average theta code look like? That's what this is. So this is the hippocampal code as a function of relative position, animal's head. And by the way, like the place cells are centered at a location. The positional estimate is best like right between their eyes. So the animal's location, if you think, where does the animal think it is? It's where its head is, right between its eyes. Like where its body position, doesn't make any difference. It's the head position angle, that's its location. And so right here, current head position, and then behind and ahead, what you see is that every theta cycle, over as a function of phase, I go from just behind to just in front of them. That's again, behind and front, which also happens to be because these place cells are not just sensitive to animal's location, but also the heading direction, if I'm moving in this direction. So behind and ahead generally means past and future. So this is where I was, this is where I'm going to be. So 10 times a second, you're doing this kind of retrospective estimation, prospective prediction. And the idea would be, okay, so that's the code that you actually see being expressed here. How are you actually getting this code? And where is it actually going to? So we're limited on time, so I'm going to cut through a lot of the details to say that what you find is that the hippocampal circuit that contributes to the generation, this is measured in CA1, the hippocampal circuit that contributes to the generation of this part of the code, the future estimator, is different than the part of the circuit that contributes to the present past state. Now, how is that and why is that? Well, interestingly, as I talked about before, CA1 gets input directly from the entorhinal cortex and also through this classic trisynaptic loop, dentate CA3. So CA3 projects into CA1, the entorhinal cortex directly projects into CA1. So CA1 has these two inputs, and these two inputs, they're both theta modulated, just like this. But interestingly, they're theta modulated, but they're phase shifted. And so when you're looking in CA1 is getting different input at this phase than it is at this phase. And it's actually getting input here at this phase largely from the entorhinal cortex. This is the direct entorhinal input. And then this phase is getting input from CA3. So you have two different circuits are contributing to both. Entorhinal present state, CA3 estimated or predicted future state. And so you could say, oh, okay, that's kind of interesting. And so you have both circuits. Well, who cares about these two things, present, future? Now, present state, that could be quite important. It's oh, am I sitting here? Is this where the owl's going to come? Okay, I want to know exactly where I am. And so you could say, oh, that's the rate-based thing. So the rate-based thing, that's where I really want to know, am I at the location, right? Right now, am I at the location that I think I am? And so that early phase rate-based code is driven by the entorhinal cortex. And then the late phase, where you get the temporal sequence, the phase precession effect, the temporal component of that is driven by CA3. And so the idea is CA3, you're actually trying to, when you're doing an estimate, it's okay, where am I right now? I'm going to, I've got this rate code. I'm pretty sure I'm where I am now. And now I'm going to do this estimate into the future. And CA3 is going to help me do that. And so CA3, now is CA3 driving the sequence? It's just contributing to it. We could talk in detail, how do you actually get this kind of sequential dependency? But the idea is it's CA3 dependent. And CA3, you can say, oh, it's doing this pattern completion. It's got all this auto-association. And so what it's really doing, it's taking partial input, it's using prior auto-associative learning to make a likely estimate. And so you have simultaneously going on behind, ahead, past, future, early, late phase. All of those things come together to point to a circuit that's capable of doing this kind of sequential estimation and prediction. But who's it talking to? As I mentioned before, prefrontal cortex was the one that's the one that tells you whether to pick up the phone or not in the right context, right? So you need that. And damage to the prefrontal cortex here, which is connected to the hippocampus, they both produce similar kinds of deficits. Episodic memory deficits, navigational deficits, but for slightly different reasons. Again, you need the hippocampus to give you the contextual, differentiation, and then you need the prefrontal cortex to do the appropriate response mapping. So if I don't know I'm in my house or my friend's house, I can't get the right response. If I know I'm in the two houses, but I can't map them into the appropriate responses, I also don't do the right thing. So I need both. And so this so-called loss of executive control. I'm just going to kind of run through all of this quickly. So this is a simple task where this kind of left-right maze choice task, it's kind of like this, doing a different thing. Same location, different thing. So I'm at the center here. Do I go left? Do I go right? Do I answer the phone? Do I not answer the phone? So I have this bifurcation in choice. This is this task where the choice depends on something else. It could be like the previous response. Alternation is typically used here. You say, oh, I go left here if I went right last time. So I use past information to determine future response. And what you find is, again, hippocampal damage, prefrontal damage, impair performance on that task. recording in both. This was that data that I showed you. What you see is that, yeah, the prefrontal cortex seems to lock to the hippocampus when an animal is performing this task, but it doesn't lock all the time. It turns out it only locks under a certain condition. And in this task, this is actually a slight variant of that T-maze alternation task. In this case, it's like back-to-back T-mazes. And there are two directions here. So the task is the animal starts in one of these two locations, and then depending on where it started from, it has to choose here. So if it started on this, like the upper arm, when it runs over here, it has to go to the upper arm. And then it turns around and it comes back, and then there's a little door here that we choose. We force the animal into one of these. So in this direction, we choose. In this direction, the animal chooses. And what you find is that, and then you look at activity in the two structures, compare spiking. There's spiking in the hippocampus. There's spiking in the prefrontal cortex. There's this theta oscillation, blah, blah, blah. And now we measure a couple of things. One, we look at the phase locking of the prefrontal cortex to the hippocampal theta rhythm. And the phase locking to the hippocampal theta rhythm occurs here at this part in the central portion of the maze, but only when the animal is going in the choice direction. It's when the animal is going this way, but not this way. But additionally, you see this coherence not only when it's going this way, but it's when it goes this way and it gets it right. If it goes in the choice direction and gets it wrong, that is indicated by the loss of coherence. So they're like the conditions. You need the coherence for the hippocampus to tell the prefrontal cortex, oh, remember we started in the left or the upper arm. And in this direction, you don't get the coherence because the prefrontal cortex doesn't care. It's like it doesn't matter, right? I don't need to listen to the hippocampus. I'm doing my own thing because I'm going to be fourth. I don't have to make a choice. And so the presence or absence of coherence here, if I were to just look at, as the animal approaches this, if I see that they're coherent, I can say, I think he's going to get this right, or he's going to get it wrong. He's not listening to the hippocampus. He's like totally 50-50 on this. So you could predict performance based upon the presence or absence of the coherence, which would serve as a channel for prediction. Now, an additional feature to this, which I didn't actually show in here, and that is, let me just go to this sort of the model. So when you kind of get to this, so like different phases carry different information, past, present, future. And it turns out there is this phase locking that goes on here, this phase locking, but different prefrontal cells have different preferred phases. So you notice that this one seems to be locking here on the, sort of on the peak. Different prefrontal cells will lock to different phases. So it's as though there are these phase channels. There's like the present, where am I? Then there's the future. So different groups of prefrontal cells could tune into different past, present, future channels. So you could have this heterogeneity, prefrontal cortex, and it could change those channels just by shifting its phase preference. And so you have this idea that you have phase coherence, but also you can change phase preference and then have dynamically either listen to and then change the channel of the hippocampus that you listen to through this dynamic phase coherence control. Now, that was all I was just like making that up, right? Sure, I see that there's this coherence, but does it really make a difference? And so I just quickly summarize this. So this is a test of that hypothesis by using optogenetic manipulation. And so the idea is we're going to use closed loop manipulation of the hippocampus. We're actually going to go and take that phase precession curve, and we're going to selectively inhibit either the early phase or the late phase. And so we can block activity. So we drive inhibitory cells, and then we can shut off excitatory cells. And we can do that by locking that to either the peak or the trough. So we're going to block activity at the early or the late phase. And then we're going to ask, does it make any difference? We're going to have the animal run exactly the same task, but we're going to add in another little twist. And that is we're going to do that not just here, but we're going to do it in 2 task phases. So we're going to fiddle with this early late phase manipulation when the animal starts, and also early late phase manipulation when the animal chooses. And when Josh Siegel, the graduate student in my lab, he did this, what we expected to find was, look, there's going to be this critical right here. It's going to be when you inhibit the late phase, the late phase, the CA3-driven predictive information. Right here, when the animal's going to make a choice, you're going to get a deficit. Okay? And then here it won't make a difference. So that's what we were expecting. Phase selective, task phase, theta phase selective deficit. What he actually got was not that. What he got was, he got this weird double dissociation. And that is, it wasn't like there was a best phase. What he found was, you actually got an improvement in performance that depended on the theta phase and the task phase. And that is, when you blocked late phase information here at the encoding segment, animal got better. When you blocked early phase, this kind of rate information in the retrieval segment, the animal got better. So you got improvement in performance when you blocked the irrelevant component of the phase. And so here, the idea is, when you block the predictive component, you think, oh, that's going to give you the deficit. What you actually get is, when you block the kind of the coding, the rate component here, the animal gets better. And you get the converse here. When you block the predictive component, you leave the encoding component, the animal gets better. So it's both phases, the encoding and the predictive, early phase and late phase, both are relevant, but they're only relevant at different task phases. Early phase is relevant here, late phase is relevant here. Now you can ask, well, why didn't you get more of the deficit? You can argue that. It's just the idea that it's not that there's a good phase and a bad phase. The prefrontal cortex seems to be attentive to both the early and late phase, just at different task phases. So the phase matters, and it also depends on the behavior. That's the basic idea. Now, you can kind of think of this, and again, what I point out is that these different the different theta phases are driven by different circuits. So this just sort of illustrates this idea that entorhinal and CA3 inputs are alternating the entorhinal CA3s. And so what you really have here, another way I could think of this is, oh, what I have here is entorhinal input, CA3 input. When I block the CA3 input here, I get better coding. When I block the entorhinal input here, I get better CA3-driven activity, right? So it's when I block the irrelevant component, I shift the emphasis onto the remaining component. And here it's CA3, and here it's entorhinal. And so it's very much like, it's like a concentration thing. If you're sitting here and talking, it's like, okay, sometimes I'm talking, sometimes you're talking to your neighbor. Suppose I'm saying something really important, and your neighbor's talking. It's like, no, please, I don't want to hear from you. CA3, neighbor-associative interactions, please shut up, I want to hear what he's saying. There could be other times, oh, it's like I'm totally, it's like uninteresting, I'm talking about stuff. So I tune you out, and now I listen to the auto-associative components. I listen to your neighbors. And so the direct input-driven, the entorhinal component dominates here. The auto-associative, the neighbor CA3-driven component dominates here. And the idea is that both, under normal conditions, both are present. This optogenetic manipulation just allowed us to selectively suppress those two, one or the other component, and then therefore led to an improvement in performance. It's like, I could optogenetically shut up your neighbor, and then you'd be able to pay attention to me better. Or could optogenetically silence me, and then you'd be able to hear your neighbor better. Well, depending on the behavioral context, you might want to do both of those, yeah.
It's a bit hard to think about kind of the normal without perturbation performance as actually worse than it could be. Because this is, you're saying that the performance improves if you silence the neurons, right?
Yeah, so you could say, why didn't I get a deficit? Really, the reason you don't get a deficit, it's more a floor effect. And that is in the task like this, when the animals they're at kind of 60 plus %, there's a lot more room for improvement than there is for deficit, going from, chance performance 50% at this point. And so it's just harder to see a deficit. So I wouldn't overinterpret the lack of deficit here as indicating that there's there's like a relative imbalance in the positive versus negative contributions of the two components. That's really more a consequence of the imbalance in the task performance diagnostics. There's sort of a floor. You're closer to 50%, so you can see more upside than downside. So I would expect that you would get, by blocking the other component, you would see more of a deficit. And then the other part, just kind of experimentally, and that is, it's sort of an imperfect, the phase, while it's, the kind of the early late phase, it's not completely blocking, it's just a biased, you're blocking more or less of the early or late phase component. So, and so this idea, you got these three things. You got sort of the phase shifted inputs, enter around, let's say A3, different circuits engaged at different theta phases at the output, and the different phases of the output coding information is differentially, reflective of past, present, or future state. And then you add into that this idea that the downstream prefrontal cortex has this preferential phase locking, that is listening to the different components. So now you can see that, well, it probably is a different subpopulation of cells here, prefrontal cells. When you actually inhibit this component versus this component, you're actually influencing which prefrontal cells are actually going to be, are going to be able to participate in the decision making. If I block these, prefrontal cells that were preferentially phase locked here are going to be kind of deprived of the entorhinal cortical input. So in a sense, I'm also influencing which prefrontal cells might be participating. We didn't really distinguish or establish exactly which of these two hypotheses was responsible for the difference in performance. That is it a bias of the hippocampal representation, or is it a downstream effect bias of the prefrontal populations that then are responsible for guiding the behavior? But an interesting property of this coherence that I had mentioned before is it just tunes in. It selectively engages right here, about 500 milliseconds, 5 theta cycles just before the choice point. That's when the prefrontal cortex actually seems to care what the hippocampus is doing. And so that does not account for, the prefrontal argument doesn't account for the effects here in the encoding segment. That would be something that, it could be like another downstream structure that's contributing that. But anyway, that's the theta, the theta story. This just kind of illustrates this preferred phase. And different prefrontal cells will have different preferred phase locking profiles. So now, the encoding of sequential information through the theta, to sort of the theta oscillation, and sort of interestingly, back to this sort of theta coding idea, there's a couple of schools of thought here. One is that there were some classical manipulations that, sort of cholinergic inhibition. So when animals are running, you see the theta oscillation. That is indicated. It occurs in the context of elevated acetylcholinergic tone. So movement is associated with elevated acetylcholine, increased attention, increased acetylcholine. Acetylcholine is associated with the theta oscillation. There was some work suggesting that you could just dump in a slice acetylcholine and drive the theta oscillation, and that's a little bit controversial, but nonetheless, it's the two are related. Acetylcholine, learning and memory. You deprive animals of acetylcholine, get learning deficits. This is like a classical manipulation. You go in, you lesion cholinergic structures, animals don't learn memory as well. You put in cholinergic antagonists, animals have poor performance. It's like acetylcholine's the memory, it's like the memory neuromodulator. You can go to GMC and buy, choline supplements, the precursor to acetylcholine, all the same arguments. Now, one argument would be, okay, so the acetylcholine kind of works by helping to, helping to drive the theta oscillation, and the theta oscillation is really the thing that you need in order to get, that's effective coding. All of these things sort of work in concert. One interesting property of the sort of acetylcholine is, so there's a variation of that. Lowering acetylcholine now gives you a transition from this theta mode to the non-theta mode that we'll talk about, this bursting activity. And so the quiet wakeful state is associated with reduced cholinergic tone. So if I do a behavioral experiment, when an animal's running in the maze, and I block cholinergic receptors, memory deficit. When the animal is asleep, if I, instead of that, let the animal run, and then during sleep, I do the same manipulation. You know, memory, it's the memory drug, man. I'm going to deprive you of the memory drug during sleep. What happens? You improve memory performance. So reduced to, okay, now I'm going to do the opposite. I say, oh, no, man. Memory drug, man. I give it to you. I'm just, you know, you give the memory drug. I give cholinergic supplement during sleep following acquisition. What do I get? I get deficits. So it's like, wait a minute here, wait a minute. And so it's very much like this, right? I thought there was like, right, acetylcholine good, you know, high levels good, low levels bad. No, it depends. It's like this double association. High levels during behavior, good. Low levels during sleep, good. So it's that. So what is associated with low levels of acetylcholine during sleep, and why would that improve performance? Well, that gets us to this sort of sleep. You go to sleep, you get reduced levels of acetylcholine. Reduced levels of acetylcholine increase the expression of that bursting activity that you saw. And so there's this idea, well, maybe something goes on during sleep that actually, you know, contributes to memory, learning and memory. And you know, we can kind of broadly identify two stages of sleep. There's non-REM sleep, largely evolutionarily conserved. It's sort of argued that it's associated with periods of immobility. They're like these invertebrate sleep models. Sorry for the scare quotes, but you know, sleep, immobility. They're really, you know, like flies sitting quietly immobile. The argument is, well, this is like, you know, the invertebrate analog of mammalian sleep, because in mammalian sleep, you not only have, there's sort of certain defining characteristics of sleep. There's both the sort of behavioral characteristic, but then there's also this idea that, you know, sleep is something that actually serves a larger function, that it's homeostatically regulated. You have a need for, it's not that you just go to sleep, you have a need for sleep. It actually does something. And if you deprive sleep, then the system or the body works hard to get more of it. So that's sort of required. So sleep deprivation, if you deprive an animal of sleep, you get sleep rebound. And so this is now sort of interesting. A lot of the invertebrate models, they sort of have indications that yes, it's not only immobile, but they also have, there seems to be like some sort of homeostatic regulation of that. And so this idea that immobility and non-REM, and during immobility, that's where you get these kind of bursts of activity. Electrophysiologically, it looks very much like non-REM sleep. And then REM sleep is sort of an interesting thing. It was believed to be like an evolutionary innovation. It was sort of like a mammalian thing, and that is that sort of, you know, mammals that had like kind of, you know, more circadian regulation of their sleep, that is sort of light-dark cycle. They would sleep, you know, either sleep in the light or the dark, that there would be extended periods of immobility. And then during those extended periods, there would be, instead of just having, you know, hours and hours of non-REM sleep, you would interrupt that. And you would interrupt that with periods of sleep. You're immobile, but the brain activity looks very much like the awake state. You see the expression of the theta rhythm, and that, you know, you sort of inhibit your neuromuscular output, so that you don't actually move, so you're paralyzed, your brain looks like it's awake. And so REM sleep, referred to as paradoxical sleep, because even though your body looks like it's asleep, your brain looks like it's awake. And so the idea is that during REM sleep, at least subjectively, this is when people, if you wake them up, they report dreaming activity, even though actually reported dream content can be found in all stages of sleep, both non-REM and REM sleep. So I'm just going to focus here, the remaining time, on this non-REM sleep, which is very much like immobility, which might be something that is conserved all the way back to flies. What I'm talking about here, I don't know, what's the farthest you can actually, I have a postdoc who's actually right now, he's here, he's doing work on frogs, and working on, I mean, they're not even invertebrates. So he's looking at these sort of jellyfish like organisms to see whether, how far back does the kind of sleep and the homeostatic regulation of sleep go. Sure, fungi, there you go. I mean, there's, yeah, okay, well, like, do we even have to, like, keep it to, eukaryotes? I don't know. I mean, it's like, I would say, and what I'll point to here is that there's sort of two aspects to the idea of homeostatic regulation of sleep. One is the metabolic idea, and that is that you just need downtime, you need, like, energy restoration. There's a lot of housekeeping functions that are required, and that makes total sense. But then there's the other side, and the other side comes, and so that's this idea that there's like a kind of a cognitive need for sleep. And this comes from the idea that once you have a nervous system, so it may very well be, yeah, like sort of sleep homeostasis, go back to, fungi, sure, because they have metabolic needs too, right? But if you have a nervous system, the idea is that once you actually kind of turn off, you kind of go into housekeeping mode, what you find in nervous systems, it's very hard to, like, have neurons that are not active, that don't generate spikes. Because when you don't generate spikes, right, it's like you got to turn off everything, man. I got pumps that are there that are, I have a lot of biochemical processes that have been tuned based upon an expectation of activity. And so, like, during these sleep or mobility periods, you don't just shut neurons off. They stay active. They might change their activity mode. And so the idea is that when you have this downtime, offline mode, where cells are still spiking, evolution doesn't waste anything. It says, look, you're here, you're generating spikes. Why don't you just make those spikes actually useful, right? If we can use that spiking activity to do something, and it provides some benefit, and so that's why this argument that you could go back to fungi, you could find this kind of homeostatic regulation of metabolic needs for sleep, but then the emergence of nervous systems said, particularly where you had, like, spiking activity, said, if you're going to generate spikes anyway, let's just make those things useful. And so that would be the argument here, that there's something useful that can be derived from spiking activity that occurs in the absence of external or sort of environmentally driven input. And so the demonstration, there was something going on during sleep. So this is like the experiment. You have an animal sleep, you have them run, you have sleep, and then you just look at activity in the two states, before and after. Is there any difference? This actually shows this sort of cartoon illustration of sleep structure is actually derived from a real sort of sleep architecture of an animal, and it illustrates one thing. And so I don't know why this is not green, but it's, the sort of the gray-green. These are periods of non-REM, largely slow-wave sleep. Slow wave refers to the slow oscillation, like around a hertz or so, of activity that's associated with this period of non-REM sleep. And then in REM, this is where you see this kind of theta modulation. So what you find is that when you first go to sleep, the first thing you enter into is this, like, immobility that transitions into non-REM. And then the longer you're in non-REM, you will now, at the end of one of these non-REM sessions, you're going to kind of drift into, there's like a little transitional arousal. There's like an arousal event that occurs here. They sometimes refer to as like microarousals that occur. And at this microarousal transition state, you either wake up or you go into REM. And so here, drifting deeper and deeper and deeper, so-called like deep sleep here and then. Deep sleep, you're coming out of it, you go into REM, have a short REM bout, and go back into it. So this is the so-called sleep cycle, from non-REM, largely slow wave, to REM, and then back again. The period of this, like the REM to non-REM cycle, can vary in humans. It could be like an hour and a half. Here you see in a mouse, it can be a matter of sort of like 20, 30 minutes. Interestingly, this idea that REM sleep was somehow a mammalian innovation was challenged. There was some really beautiful work by Gilles Laurent where he was actually looking in these sort of lizards and found that you have this REM, there's this REM cycling. In the REM cycling, it was like 89.5 seconds. It was like, it was very precise, short and precise. And so the lizard was very rapidly switching between these. It had these brief REM bouts, but this was like very regular. And so the idea that you have like these brief REM episodes, extended non-REM episodes, and then there's the broader, like this sort of the circadian regulation from sleep onset here. Animal's going to sleep, okay, like 8 hours later. It's just about, this is like the period of sleep offset. It's like 7 o'clock in the morning when we shift their cycle, like for us. And just before you wake up, what you see is it tends to be REM heavy. So over the course of a night's sleep, you start off with lots of non-REM, and then you end it with lots of REM. And that's the general sleep architecture. The REM episodes are more frequent, and they're longer. And so you can say, so what's going on during the non-REM and the REM? We'll focus on the non-REM. And in the non-REM state, and so this is the idea, animals runs, you look at the sleep afterward, and this was some work by Albert Lee when he was in the lab. What he noticed that during the period of sleep following running, you found these brief episodes, about 100, 200 milliseconds, where you would see an expression of sequences of activity. Now, prior to this, I had shown that during this period of sleep afterward, cells, the coactivity pattern that was seen here was re-expressed here, and that is the correlational structure of activity during run was expressed during sleep. What Albert found was it's not just the correlational the correlational structure of cells, like the place coding, but it was the sequential coding, like the phase coding that you see. So that you see these sequences being expressed. And those sequences, it turned out, when you see those sequences, they co-occur with these so-called sharp wave ripple events. Here, this is the ripple part. The sharp wave has been removed, has been filtered out just for ease of visualization. But this is one of these sharp wave and then ripple events. And on top of that, you see one of these sequences. Now, there is something here. So in the paper, we kind of talk about sharp wave ripples, the expression of sequences, and they kind of fit with this model, the previous model I talked about, the phase precession model. But there was another property here that we hadn't even thought of, and it's in the figure, and that is there's this longer sequence, and this is actually 2 sharp wave ripples. This is not a single event. There's actually 2 events. And so this anticipated a subsequent result in which we found that longer sequences, if you force an animal to run on a longer maze where you get longer sequences, what you find is long sequences are expressed by sequences of sharp wave ripples. So you kind of chain together multiple events like this. And so there's this idea that you compose longer sequences with shorter sequences. Now we kind of get back to, well, the idea that you compose, that there might be some sort of elemental components, like a sharp wave ripple, that you can kind of flexibly assemble into sequences that you actually then use for what? Okay, maybe it's this downstream training, systems consolidation. It's like, again, we got our chat GPT network somewhere, our hallucinating generative AI. Yep.
Is there some knowledge about what's the chronology of these replay events? We replay first the most significant memories from the world during the day, or is it in the chronological time it happened?
Right, so chronology is there. So if you kind of look at where do these sequences derive from? Like over the day, we actually did some experiments looking at that. If you have animals experiencing multiple events over the course of a day, what do the replayed sequences look like following that? And what you find is that immediately following, one session, there's a period during the non-REM sleep with a time constant of, maybe an hour or two, where you see a very high likelihood of expression of the recent events. You run them in a new maze, they go to sleep. The new maze tends to get expressed as well. But you also find that there is an interaction between recency and the relative familiarity or novelty. And that is, so let's suppose you do, maze 1, maze 2, and you just kind of repeat this maze 1, maze 2. And now you introduce a new maze, something new. Now, what would the logic be? What would your expectation if you introduce a new maze, and now you look at the sleep immediately following that? And that is, one thing about this effect, the reactivation effect here, and that is that if you do this repeatedly, there seems to be a novelty interaction, and that is that it's things that are kind of novel. They sort of change properties of these replayed events. but not necessarily whether they occur, but how they occur. And in particular, I don't know if we'll get to it, even though I've illustrated these sequences in this kind of forward-reverse thing that I showed you in the theta, like theta phase precession, and these basically look like the same sequences during theta phase precession. It's also possible for these sequences to be expressed in time reverse fashion. This was so-called reverse replay that was discovered by Dave Foster when he was a postdoc in the lab. And what Dave found was that when you put an animal into a new environment, and the first time, even when they stop, and also during sleep, you find an increase in the incidence of these reverse replay events. And so the direction of replay can be influenced by the relative novelty. It's like, wow, this is something it's really new. Maybe it's like something I need to learn. And so you get more of these reverse events. And so the idea with the reverse events, and we'll kind of get into this, they sort of fit into models of reinforcement learning where, it can help contribute to resolution of temporal credit assignment. But back to this, which environments get expressed? When you introduce, if you had like multiple previous things and you introduce a new environment, you increase the frequency, so you get more replay events, but you don't get a bias. It's not that more novel events. You actually get an increase in incidence of familiar events. So you increase, it's like I increase overall sleep processing, but it includes both recent and prior events. And so the idea that novelty, when I have something new, it just like turns up, says, sleep, man, we need to work on this. But that everything gets bumped up, and it just tends to, you're gonna have more non-recent events than recent events. And so it gives the appearance that novelty actually drives an increase in the likelihood of expressing non-recent events.
And since there's really both short events and the long.
Yeah, that's a great question. The short, long events, there was some relatively recent work. So Lauren Frank, who's also a graduate student who did and Yuri Busaki also looked at this, found that the incidence of long replay events was actually associated with improved performance. So the long events are actually somehow sort of more functionally beneficial. Now it is the case, so when you go in and you just do the disruption, if you block these events, you go in, closed loop disruption, you impair performance. So you slow down the acquisition basically if you interfere with these events. Now why would long events be better than short events? I think that there's, that's kind of a lunchtime thing. We can kind of talk about that. But all of those things, whether they occur, the direction that they occur, and the length of these, particular multiple sharp wave ripple events, all very likely contribute to whatever the computational imperative or benefit, that's derived from this sleep-related activity. And then if you look in the little time, but if you now look in the neocortex, and so I had mentioned to you, okay, the hippocampus is also like communicating downstream. Now there is the direct communication is really with the retrosplenial cortex, but then retrosplenial cortex can kind of talk to a lot of other structures, including adjacent visual cortex. So Diane G, when he was a postdoc in the lab, did this experiment recording the hippocampus and the visual cortex, primary and secondary visual cortex, and found a couple of interesting things. One is actually you get place-like fields in the visual cortex. So this is, rats running around. This is a visual cortical cell. They fire in a location, and it's not strictly dependent on the visual cues. I mean, there's obviously some visual, input that can modulate their activity, but where they fire seems to be more influenced by location than the actual cues that are present at that location. And the way you can demonstrate this, you just shut the lights off, you turn off the lights, you get about a sort of tenfold drop. The firing rate goes down, but the location they fire remains the same. So there's like this conserved locational firing. And so again, you can think of these two properties, where they fire and the firing rate. You see the same thing in the hippocampus. In fact, we were talking during the break about, I had forgotten to mention, like these remapping things where you fire, do I fire here in the morning or in the afternoon? There are really two kinds of remapping that people generally refer to. One's called global remapping, complete reconfiguration of the cells. If you sit there versus over there, you're gonna get a completely different set of cells that are active. But like if you sit there over time, or if I if I introduce, I like introduce shocks into your seat, you're going to get the same cells fire, but I'm going to modulate the firing rate. So firing rate, referred to as rate remapping. So changes in firing rate often accompany changes in cues or configurations of an otherwise static context. So I need to do both. I need to know, it's not like, someone moved the chair, it's a whole different room. It's like, they moved the chair, it's the same room, but the chair has changed. So global remapping, rate remapping. And so what you find here, like in the visual cortex, I change the cues. The conventional wisdom is, like the mapping, the cues are the mapping. So it's global remapping, right? If I change the cues, obviously I would change the visual cortical output. Well, if they're moving in space, you actually have this positional code, and then you get rate mapping of the cues at that location. So he found that was in the visual cortex. And then additionally, when the animals go to sleep, and so that what that means is that while the animals are running, both in the hippocampus and the visual cortex, you get this sequence. You can see this sequential activation in different cells. So you could do the same kind of positional decoding with the visual cortex as you could with the hippocampus. And when you do the positional decoding, you find periods of sleep where you get sequences, hippocampal sequence, visual cortical sequences, and the two are actually coordinated. But they're coordinated in an interesting way, and that is that there are these periods, what I refer to of slow wave and slow wave sleep, these so-called slow oscillations, about 1 hertz, where you find periods, activity, inactivity. Now, when you look in isolation, if you look at individual cells, these periods of activity and inactivity are associated with switches in so-called up and down states. They're periods where cells are depolarized, you get lots of spikes, and they get shut off, hyperpolarized, on, off. And these kind of alternate. You do it across a population, all on, all off. This is the slow wave. And then you look at the slow oscillation in the visual cortex and the hippocampus, and you see something interesting. The two are kind of coordinated, but you notice it's like the neocortex turns on just a little bit before the hippocampus. So you see these slow oscillations. But in the hippocampus, unlike the neocortex, if I take one of these neocortical cells out in isolation, I can get spontaneous up and down state. The cells themselves express up and down states. Hippocampal cells don't do that. So you don't get spontaneous up and down state. This activity is driven by the neocortex. And so the idea is that during slow wave sleep and during these up-like states in the hippocampus, this is when you're getting sharp wave ripples. This is when you're getting the replay events. And so the idea is that these up-like states in the hippocampus are driven by activity from the neocortex. The neocortex turns on, it drives the hippocampus. The hippocampus takes those inputs from the neocortex and starts generating sequences. And I say generating because that's probably what it is. Like the neocortex says, remember that stripy pattern? So stripy pattern, and the hippocampus says, stripy pattern, that was at the corner of the maze, and you kind of ran around there, and you ended up in the, with the dot pattern. And so you go from like a static sequence here, initiation of a static state, hippocampus starts to express sequences. So the neocortex leads the initiation of these ripple events. But then when you look at the sequences in the neocortex, it lags the hippocampus. So this starts it off, but the sequences in the hippocampus lead the sequences in the neocortex. So you have this like back and forth. Neocortex starts, hippocampus expresses it, it drives sequential activity in the neocortex. And this is actually kind of a good example where you see the whole thing is shifted. Neocortex starts, hippocampus starts generating sequences, and then the neocortical sequence terminates a little bit before here. So the hippocampus runs on a little bit, and it has the opportunity to influence the next neocortical upstate. And so it's like this back and forth, both within, neocortex starts, hippocampal sequences driving neocortical sequences, and then hippocampal sequence able to kind of trigger or bias the next upstate. And so now there's this longer organization, which is the longer organization. If I wake you up and ask you, what were you dreaming about? This is probably what you're going to be telling me about. The detailed content was probably here, but this longer organization, the sort of the narrative-like organization, is probably expressed here in this longer time scale. So, we're basically out of time. But I just want to point out, so this idea that the neocortex actually biases the replay of detailed hippocampal replay sequences could be easily tested, and we did test this by just biasing the neocortex. And you could take advantage of the fact that, in particular, the auditory cortex remains vigilant during sleep, and that is that its response to sound is unchanged from awake to sleep state. That's probably an evolutionary, feature, and that is during sleep you're vulnerable, but you need to be attentive. Like if you hear something, and the only reason you don't generally wake up, I like to point out, during sleep when you hear, because you're hearing everything, it's not something that you need to respond to. If you hear the TV going on, it's like, look, it's just the TV, I don't need to hear it. If it's like the background noise from from your noise generator, it's fine. I hear it, but I'm not going to get up. If it's something unexpected, that it's like if I call your name out, you can wake up because it's something that that you might want to respond to. So, auditory cortex is it's hearing everything during sleep. Auditory cortex awake, sort of. Hippocampus asleep. And so if you just bias, so train an animal task, it hears a sound, one sound goes left, another sound goes right. Simple auditory association, spatial association task. Animal goes to sleep, now you start playing the left or right sound. See what happens. And when you play the left sound, so here with the animal running the task, you get some place cells that are on the right side, some place cells that are on the left side. This is just the decoding during the task. Place cells on the right side, place on the left side. And then the animal goes to sleep. When the animal's asleep, you see that you kind of switch from, this is the activity during wakefulness, like here's a place cell that's active at the location, the animal goes to sleep, and now you're starting to get these like bursts of activity. Here, you just like zoom in on this thing, you see this is one of these sharp wave, these kind of slow oscillation like sharp wave ripple burst events indicated by elevated multi-unit activity. You decode the precise, here you see like there's a sequence being expressed here. And so you see there's content, decodable content. And so we're just going to take this decodable content that consists of left side and right side. We're just going to ask, can we influence the decodable content based on the sound? And the answer is, yeah. When you play the left sound, you get activate cells in the left place field, play the right sound, you get sounds on the right place field. Now what's interesting in this, I don't know if I have is that the sound in this case was played just briefly, like 750 milliseconds, but the bias persisted for like 10 seconds. And so the idea is you like trigger it, and then the bias content persisted until you played a new sound, changed the bias, and now you get like the new bias. And so there's the idea is there's like this kind of reverberation. Neocortex triggers, you can trigger a sequence, and then there's some kind of iterative processing that continues to go on until there's some kind of change in neocortical state. Now, and so the idea that this is how something's being trained and that it can be influenced by the sensory cortex. And this was actually demonstrated, a fun demonstration of this was done here, very similar to the experiment I just described. It's just that it was done psychophysically, and here, these are people that are trained on a simple, like, a spatial matching task. It's like if you play this, you got these cards that are laid down, right? You turn them over and you see, oh, like the cat is over here. So different cards, different images, different locations. And you just have to remember the location of the cards. And what they did in this experiment, this is in Ken Paler's lab at Northwestern, they added an additional twist. When you flip the thing over, when you flip over the cards, not only do you see the cue, but you hear a cue associated. So they would play a sound, cat sound, teapot sound. And then what they could do is during sleep, they could play one of these. And so if they played the cat sound, when people woke up, they had better memory of the cat locations. So not only were they able to improve performance through manipulation of sleep, but they were able to improve performance selectively on the cue content that had been introduced during sleep. So you could improve bias by, presumably, when you played the cat sound here, what happened is the hippocampus starts like driving out cat-related, memories, cat location, and then that improved subsequent performance. And so it's just the simple idea, you kind of cortex biases replay, replay, transfer, I'm not going to take ownership of that, even though I put it on this slide. It's like transfer is very weak. It's the idea that there is some influence of the hippocampus on cortical representations that then subsequently improves performance. With that, I'm just going to, I was going to kind of show this reverse replay. And so, everything I talked about, we kind of see, oh yeah, it's like playing back memories. But what Dave Foster discovered, and this was like, he's running an animal in the very same task. You just put an animal on a maze track, animal runs one side of the track, runs to the other side. And the very first time it does that, generally, animals they're not like super performers. Maybe they'll do it once, and then they kind of stop. In this case, first time, animal runs to one side, and then he just sits there for, it's like 10 minutes, he's just sitting there. And during that time, if you actually look at activity that occurs, what Dave found was you see these sort of burst, sharp wave ripple sequence events, but in this case, they occurred in reverse time order. That is, they started from where the animal was and they ran back onto the maze, these so-called reverse time sequences. And this was interesting because Dave Foster, he had actually done his PhD work on reinforcement learning and how you could use the hippocampus to do reinforcement learning. One of the problems in just like sort of basic reinforcement learning, so-called temporal credit assignment problem, trial and error learning, where you get like one shot, you get a reward. You really want to you want to reinforce events that preceded or led up to the reward, not just the reward location. And what he noticed is this would actually be a pretty good mechanism. Reverse replay paired with a dopamine reward signal would be a pretty good mechanism for one-shot temporal credit assignment learning. We're out of time, but I will sort of point out that we did this experiment to actually record in the VTA to look at dopamine signals, and what you find is that indeed, paired with these replay events, not just reverse replay, but with these replay events, you see associated dopamine signaling. So there is reward signaling associated with hippocampal replay. And so the idea that there is some kind of reinforcement learning mechanism going on during these events seemed to be supported. But Foster went on to show that it's not just, so these replay events, not just reverse replay that can be predictive of improved performance, but that even the forward replay events seem to be biased and, during active goal seeking, seem to be biased toward goal locations. What does that mean? We don't have enough time to kind of go through all this, but the idea that both forward and reverse sequences could contribute to learning, they could contribute in different ways. Forward, reverse, sort of planning, reinforcement learning, and then this is just kind of showing, kind of elaborating on that point that I made earlier, and that is that long sequences, if you have an animal on a long track like the one I showed you before, those replay sequences are actually, they take the form of multiple sharp wave ripple bursts, and they come in this kind of segmented, or what I would argue is likely to be some kind of compositional representation, where the elements that are actually expressed during individual sharp wave ripple events are probably constructed in order to serve in the service of some kind of compositional learning, which in the case of spatial learning, the compositional elements are basically just the spatial elements. But I think if you were to introduce some kind of multi-dimensional learning where you have, kind of multiple, kind of selective interactions between these various dimensions, and that those interactions could be, kind of compartmentalized or restricted to certain regions of space, the idea is that you would learn those regions of space where you found these kinds of local interactions, and that those are the things that would be expressed and then potentially could be flexibly reassembled here. So I think that's it. Just this last point, the sharp wave ripple sequences, when you look at sharp wave ripple sequences, these sharp wave ripple sequences are basically the same. They're on the same spatial scale and the same temporal scale as theta sequences. And so the idea is that the theta sequence is the compositional elements that are actually captured during theta. So during theta, you're actually kind of learning what are the the kind of local temporal dependencies. And again, I kind of point back to, it's like, the transformer models where essentially what you're doing is you have representations that are capturing local time structure, mapping that into elements. Now to say that those are actually, that the underlying learning that goes on is some kind of hidden compositional, has some hidden compositional structure, I'll let all the machine learning dudes, and women too, not just guys, figure that out. Because it's likely, figuring out what are optimal representations, how can the architecture be tuned to facilitate that, and I sort of point to this organization, the longitudinal, the transverse, the lamellar organization, the kind of parallel processing, separating the direct and the indirect, autoassociative, the entorhinal. All of these are elements of the biological network that evolution has figured out generally work to solve the kinds of problems that occur in real-world interaction with limited samples as n goes to 0. And so I think that's for me, that's the kind of lesson that there are a lot of hidden properties in the biology that have been conserved that I think can be instructive and informative in thinking about AI and machine learning. And I don't think there's anything, this is just the reward activation, just showing you get dopamine activity, VTA activity. Now you just get to the end here. You get VTA activity, so dopamine activity that's selectively associated with the end, when the animals are replaying sequences that end in a goal location, you get dopamine signaling. But interestingly, you get that dopamine signaling when the animals are trained in a task where they have to make a choice. And if you just have them running back and forth on a maze like this, then you don't really get that, kind of strong modulation. And so the idea is, just like in the theta coherence effect, where the hippocampus and the prefrontal cortex only talk when the animals actually have to make a choice, similarly here, the hippocampus and the VTA only seem to talk when the animals are replaying memories of events that actually were associated with choice. And so that's the... And I'm just going to close with, this is an experiment, not one that I did, but I love this experiment. This is an experiment by Karim Ben-Shanaid, who was in Paris, not just at the Olympics, but also here doing some old school. This is my sort of bad neuroscience pun joke. An old school experiment, which is doing closed loop electrical stimulation of the medial forebrain bundle. Now, does anybody know why that's a bad neuroscience joke? Old school experiment? It's because Jim Olds, when he was at Caltech, was the first to discover that medial forebrain bundle stimulation could drive reinforcement learning. And so that is that you had animals, they would sort of put electrodes in medial forebrain bundle, they would press a lever to get electrical stimulation, and these rats would press that lever until they died. They would take bar pressing over food and drink, anything. And so this is just like this very, very powerful demonstration. And the medial forebrain bundle is the fiber system that connects the VTA to the nucleus accumbens, the VTA being these dopamine projections. So the idea is that you could electrically trigger the release of dopamine. And when you do that, that's like super addictive, which of course every drug of addiction has already figured out. They all tap into exactly the system. So anyway, old school experiment, medial forebrain bundle stimulation. If you do this, to demonstrate this, one of the ways to do this is so-called place preference learning. Animal walks along a little maze, little box, and when it gets to a certain location of the box, you just stimulate the medial forebrain bundle. This is so straightforward. We actually do this, or we did do this, as it was an undergraduate lab, a neuroscience lab, where you could just demonstrate, oh look, you can get animals to do anything with using this medial forebrain bundle stimulation. So this is the classical demonstration. Animals go to a location, you stimulate, you find they learn to prefer that location. Ben-Shanaid did a little twist on this. Animal just put in a box, wanders around, no medial forebrain bundle stimulation, but you record place cell activity. And so you record place cell activity. For instance, here, there's place cells that are active, in the lower part of the box. And then during sleep, When that place cell was active during sleep, then you stimulate the medial forebrain bundle. So he's stimulating dopamine release during sleep tied to the activation of a place cell. And then the sort of the mind-boggling thing is when you do this, and then you take the animal out and you put him into the box, animal's like, man, I love this part of the, I have no idea why. Had it never been paired with reward during wakefulness, had trained new learning during sleep. It had learned to prefer this location, even though it had never experienced reward during waking. Only during sleep, and only as a consequence of the activation of a place cell in the hippocampus during sleep. And so, to me, this is sort of the most direct demonstration that yes, learning can go on during sleep. Now, what's interesting about this result, other than the fact that it's like a very compelling demonstration of the capacity for sleep learning, and that's when we looked at that dopamine, like all that, like that dopamine activity that you saw, I showed you associated with replay. That was done during quiet wakefulness. That was when animals were stopping. When we look during sleep, you find that there are these same replay events, but when you look at the association with dopamine signaling, there's actually a negative correlation. Dopamine signaling goes down during the expression of explicit replay events during sleep. So it's as though the brain is saying, you know, there's a lot of powerful potential for learning, but I'm going to be replaying some crazy stuff here, right? I'm going to be exploring the space to, right? It's not reinforcement learning. I'm not trying to just replay the same thing over and over to like consolidate it. I'm going to be doing some other things, and during this exploration, I'm going to shut off dopamine because I don't want to be doing reinforcement learning. And so it's this idea that you got to replay in the service of reinforcement learning or some other form contributing to learning that does not actually require, in fact, could be impaired in some way or overridden by the presence of dopamine. Because, does this stuff occur? Do you actually learn something that, that you had never experienced during wakefulness, during sleep? I suppose it could, but, that probably, it's probably something that, requires like therapy afterward to undo. That this is probably a pathological condition that might be associated with altered sort of neuromodulatory tone. So that's the end of it. All right.